Hey guys, welcome to the video on protein synthesis. If you could find the part in your notes that is labeled protein synthesis, we're going to get started there. So just follow along in your notes and fill out as we go, drawing things as we go, as we are going to be doing different drawing quizzes later this week. So let's start talking about what this picture is. Okay, currently this is my picture of a cell. So the outside membrane here is the cell. And then here we have our nucleus. And then this is the ribosome that is found in the cytoplasm of the cell. And then I have a Golgi apparatus over here. Okay, so when we talk about protein synthesis or how our bodies make protein, we tell two parts to the story of transcription and the story of translation. Transcription occurs inside the nucleus and then is passed to the outside, and then translation is only happening in the cytoplasm of the cell. Both parts are going to be making mRNA. All right, so we're going to draw on this picture in a bit, but before we do, let's start talking about what is transcription. So to make proteins, we start with step one, which is transcription, DNA to mRNA, so messenger RNA. All right, this happens inside the nucleus. So step one, what we have to do is we have to unwind our DNA with something called RNA polymerase. RNA polymerase, or polymerase in general, usually helps unwind and unzip DNA from a double helix because it's very hard to replicate if they're stuck together. So what we want to do is we want to break the bonds between the C's and G's and A's and T's. So there it is, breaking bonds right here, broken the bonds, and now we have two sides to our DNA that is exposed. So transcription in the nucleus, translation outside the nucleus. So once we have unwound our DNA from our double helix, we now have two sides to our DNA that we are going to use one side to replicate and make mRNA. So again, DNA to mRNA, step two. The polymerase then takes and adds nucleotides to match to the DNA. Now, we only use one side of the DNA to code, so we're not looking at both sides. We're only using one strand. So we call this the coding strand right down here. DNA can use either or, but it just chooses one. As DNA then is unwound and polymerase comes along and starts adding, you'll see that anytime it matches with a T, there's still an A, but anytime there's an A, we now match with a U. mRNA does not use T's, they use U's, which is called uracil. So it goes through and continues to add and continues to add and continues to add until we have mRNA. So now inside the nucleus, we have developed an mRNA, and we need to take that mRNA right here and we need to move it to this guy right here, which is the ribosome. This is the start of translation. Now, remember, I talked about DNA being the master plan. It's the master blueprint. We don't want to remove it from anywhere. So what happens is once mRNA leaves, DNA ends up winding back up into its double helix. So now we're in step two, which is translation. We're going to take that mRNA and we are going to make a protein. So it leaves the nucleus and heads over here to the ribosome. So mRNA enters the ribosome. So when it enters, it lines itself up with these pockets, okay? And what it is it's allowing these pockets to read the strands of mRNA that are complementary to the DNA, okay? So what it's doing is it's reading AUG because it's going to go searching to find a matcher to AUG. That matcher is called tRNA or transfer RNA. So we use the codon to find tRNA that has the proper amino acid to it, okay? Now, codons are just part of the terminology that we use for those three triplets of DNA. So AUG is what we would call a codon. Now, this codon has something that we would call an anticodon that attaches to it. Really, it's just its matcher. So anticodons would be things like UAC. So we read the first little bit here. Okay, we go and we match the anticodon. So the anticodon is UAC. Now the anticodon's job is just to be a carrier. It's there to help this amino acid up here attach itself in the ribosome. like so. How did I get this word start? Where did that come from? All I did is I went to my protein synthesis chart that I showed you guys the other day. It's the amino acids chart. And I looked up AUG. When I looked up AUG, I found the codon for start. There's my anticodon right there. All right. So in translation, we now have started to make a protein. So now we need to continue the protein making by looking at the next mRNA and matching it with its tRNA. So now I have CGU. The matching anticodon is GCA. But again, this is just a carrier. It does nothing except match up with my codon. How did I get arginine? That is this amino acid right here. I went back to my chart. I looked up CGU, and on it, it told me it was arginine. So again, these are carriers. That's why they're called transfer RNA. They're not doing anything other than transferring the amino acid to the ribosome. So with each amino acid that we end up adding on the top, it starts to build a chain of proteins. So you can see they detach and then bond to each other. So now we're starting to make a protein chain, which will eventually wind up and build up into things like hormones, antibodies, enzymes, your muscles, things like that. We continue step 2 through 4. So using codons to find tRNA to get the proper amino acid and keep matching. We do that until all of our chain is used up or we reach something called a stop codon. So here we go. We matched asparagine using AAC, brought it over with the UUG. It goes, attaches itself to the amino acid chain. We're building a protein. And then everything shifts over so we can read the next codon, UAA. And again, we now reached our stop codon, and there is our protein. So this protein is made up of amino acids. This protein needs to figure out what it's going to do now. So now it gets packaged up and sent to the Golgi apparatus. Okay, and that is the process of protein synthesis. So I went through this really fast. So I want you to watch this video more than once to make sure you're understanding what is happening. We're essentially starting with transcription, where I'm just unwinding my DNA, building an mRNA complementary strand. That mRNA leaves the nucleus, heads over to the ribosome, where it starts to be processed to make a protein. How it's processed, the mRNA is read. We use that amino acid chart, find our amino acid that matches our mRNA, and we start building this chain. Now, you have a picture that looks very similar to this in your notes. I would like you, after you watch the video, to draw what you think the picture should be looking like. Use my example that I have here, okay? But essentially I want you to draw transcription, then I want you to draw translation. We're gonna go over it and really talk about it more in depth, but I want those notes to be filled in so you have basic understanding. So I hope you have a great day, and I will see you later.